आज हम MP सेट 2022 लाइफ साइंस क्वेश्चन पेपर डिस्कस करेंगे. सो लेट्स सी व्हाट इज द क्वेश्चन इन द पेपर. Which chromatography technique is most appropriate for purification of cloned his tagged proteins? So cloned his tagged proteins means है कि cloned histidine tagged proteins. सो इसका आंसर जो है वो है एफिनिटी क्रोमेटोग्राफी. और एफिनिटी क्रोमेटोग्राफी इसका आंसर क्यों है इसको हम डिस्कस कर लेते हैं. In this technique, because in affinity chromatography a column is packed with resin. These resin contain a ligand, and when the protein lysate is passed through the column which contains the tagged histidine proteins, they selectively bind to the resin, and while all other proteins which do not contain these histidine tags, they pass through the column. So we get after the procedure we get in the resin plus ligand plus histidine tagged protein. Okay, so the tagged protein is further eluted from the column under controlled conditions, so we get a purified sample. Okay, so selectively binding चाहिए cloned histidine tagged protein के लिए, that is why affinity chromatography is used. बाकी chromatography क्यों इसका आंसर नहीं थे हम इसको भी डिस्कस कर लेते हैं. कैटायन एक्सचेंज क्रोमेटोग्राफी जैसे है तो इसमें क्या होता है. तो कैटायन एक्सचेंज क्रोमेटोग्राफी सेपरेट्स मॉलिक्यूल बेस्ड ऑन देयर नेट सरफेस चार्ज. सो इन दिस टेक्निक अगेन वी आर ड्राइंग द कॉलम. इन दिस टेक्निक द कॉलम contain stationary phase containing negatively charged functional groups. So for example sulfonate or carboxylic group. So these are negatively charged functional group which are attached to some solid support in the column as a stationary phase, and positively charged molecules. So here everything is negative, and positively charged molecules such as protein containing positive functional group binds when passed through the column, binds to the negatively charged solid stationary phase molecules, and all the positively charged molecules are eluted, and while negatively charged molecules and neutral molecules pass through the column, only positive gets bind to the stationary phase. So after the procedure or experiment, elution is achieved by increasing the ionic strength of the mobile phase. So the elution is done by the increasing the ionic strength of the mobile phase, which further competes with the positively charged molecules for binding site on the stationary phase, thus releasing them from the column. So this happens in the negatively charged this happens in the cation exchange chromatography. Now next we talk about the anion exchange chromatography. In this the chromatography assumes similar principle such as cation exchange chromatography, but it separates molecule based on their net negative charge. यहाँ पे net negative charge के basis पे होगा, यहाँ पे net positive charge के basis पे elution या क्या हो रहा था, separation हो रहा था. तो in this also, because this is anion exchange chromatography, so here the resin in the stationary phase will contain positively charged functional groups, and the protein or the sample containing negatively charged molecules will bind to the resin, and other positively charged or neutral molecule will be passed through without binding to the resin molecule. So further, after the experiment, elution will be again done by achieving the increasing the ionic strength of the mobile phase, now which competes with the negatively charged molecules for binding. तो यहाँ पे नेगेटिव है आपके सैम्पल में, पॉजिटिव है आपके रेजन में. तो जो फंक्शनल ग्रुप जो सेपरेशन हो रहा है वो नेट नेगेटिव चार्ज के बेसिस पे हो रहा है. और अगर कैटायन में देख रहे थे तो वहाँ पे कॉलम में नेगेटिव था और सैम्पल में पॉजिटिव था. ओके, नाउ कम टू जेल फिल्ट्रेशन क्रोमेटोग्राफी. तो जेल फिल्ट्रेशन क्रोमेटोग्राफी में क्या था? जेल फिल्ट्रेशन एज द नेम सजेस्ट, यहाँ पे जेल से रिलेटेड, बिकॉज हियर द रोल इज जेल इज़ वेरी इंपॉर्टेंट. जेल फिल्ट्रेशन क्रोमेटोग्राफी सेपरेट मॉलिक्यूल्स बेस्ड ऑन देयर साइज ऑफ, या तो साइज के बेसिस पे या मॉलिक्यूलर weight. So in this technique, a column is packed with porous beads. Okay, porous beads, and which is made up of cross-linked agarose or polyacrylamide gel, and the pore size is definite, with definite pore size. So when the smaller molecules enters the pore of the beads, assume these are pores, so these smaller molecule take longer to traverse the column due to their interaction with the stationary phase. They will stuck in the pores because the size is small, but the larger molecule cannot enter the pores and therefore pass through the column more quickly. So as a result, molecules are eluted from the column in order of decreasing molecular weight. That means larger molecule will be eluted first and the smaller molecule eluted later. So first will be larger molecule, then will be smaller molecules. Okay, so the gel filtration chromatography is often used for desalting, buffer exchange, and purification of proteins based on their size. Next question is, whole chromosome paint analysis is commonly done by option colony assay, flow cytometry, fluorescence in situ hybridization, and ELISA. So the answer of this should be fluorescence in situ hybridization, which is also called FISH. So why the answer is FISH? Because in this technique the labeling of entire chromosome or spatial chromosomal region is done with fluorescent probe. So for example, this is your chromosome and each chromosome contains some locations where DNA is located, and for example we have to target this region, so the fluorescent probe is created and it gets hybridized to this region. So when the region fluoresces, it can be visualized and analyzed under a fluorescent microscope. So what is the main principle behind this FISH? It is a molecular cytogenetic technique which used to visualize and map the location of specific DNA sequence on chromosome. So the principle is hybridization of fluorescently labeled DNA probe to the complementary target sequence within the chromosome. So what is the procedure? So this was the principle, but what is the procedure of fish? In fish, cells or tissue are fixed on a slide, okay, and treated to denature the DNA. Why denaturing is done? Because when DNA is denatured, it unwinds. So it allows the probes to hybridize to their complementary sequence. So hybridization occurs, and then because hybridization of probe includes fluorescent dyes also, the probe is labeled with the fluorescent dyes. So whenever it is excited by the specific wavelength of light, it emits light from that specific region. So after hybridization, the slide is washed to remove the unbound probes, and the sample is examined using a fluorescent microscope to visualize the fluorescent signals on the chromosome. ठीक है. सो इसमें क्या हुआ, क्रोमोसोम का एक स्पेसिफिक रीजन जिसको टारगेट करना है, उसको स्पेसिफिक प्रोब कंटेनिंग द फ्लोरेसेंट डाई उसको हाइब्रिडाइज करते हैं, एंड देन फ्लोरेसेंट माइक्रोस्कोप में देखते हैं तो वो पोर्शन क्या करेगा, ग्लो करेगा. तो इस तरह से इसमें क्या हो रहा है, क्रोमोसोम इज़ पेंटेड. दैट इज़ वाई क्रोमोसोम पेंट एनालिसिस एंड होल क्रोमोसोम, यानी कि पूरे क्रोमोसोम का जो पेंट एनालिसिस है वो हम फिश से कर सकते हैं. अब क्वेश्चन ये बनता है कि फ़िश तो ठीक है, बट बाकी ऑप्शंस इसके आंसर क्यों नहीं है? क्योंकि क्वेश्चन हमेशा सेम नहीं आएगा, हो सकता है चेंज कर दिया जाए, तो हमें सब जानना ज़रूरी है. तो सबसे पहले अब हम फ्लो साइटोमेट्री समझते हैं. फ्लो साइटोमेट्री इज़ अ टेक्निक यूज टू एनालाइज एंड सॉर्ट individual cells based on their physical and chemical characteristics. So it involves passage of cells in a fluid suspension through a laser beam. So if there is a laser beam, so sample is passed through, this sample is passed through the laser beam. It contains cell or tissue, your targeted tissue, and in the fluid suspension. So when it passes, laser beam scans the whole tissue for the properties of the cells such as size, granularity, and fluorescence intensity, and the laser beam, it scatters the light and emits fluorescence, which is detected by the photo multiplier tubes. So the signal is then analyzed to determine various parameters of the cell such as cell size, complexity, expression of specific markers. तो इसका यूज़ कहाँ पे होगा? इम्यूनोलॉजी में है, ऑन्कोलॉजी में इसका यूज़ है. फॉर एग्जांपल अगर आपके पास सेल्स हैं, उसमें एपोप्टोटिक सेल्स देखनी है तो उनका अलग सेल साइज़ होगा, या कॉम्प्लेक्सिटी होगी, या नेचर होगा, तो वी कैन सॉर्ट आउट दोज सेल्स फ्रॉम द टिश्यू ऑन द बेसिस ऑफ फ्लो साइटोमेट्री. हम सेल साइकिल एनालिसिस कर सकते हैं, क्योंकि फॉर एग्जांपल अगर कोई सेल M1 फेज़ में है तो सब उस सारी M1 फेज़ वाली सेल का एक ही कैरेक्टर होगा, तो वो अलग सॉर्ट होंगी, और अगर कोई सेल G1 फेज़ में है तो उसका अलग नेचर होगा. इस तरह से सेल साइकिल एनालिसिस में ये टेक्निक यूज़ की जा सकती है. तो ये था फ्लो साइटोमेट्री. अब नेक्स्ट है कॉलोनी एसे, विच इज़ ऑल्सो कॉल्ड कॉलोनी फॉर्मिक यूनिट एसे. तो कॉलोनी एसे इज़ अ टेक्निक यूज टू क्वान्टिफाई द नंबर ऑफ वायबल सेल्स capable of forming colonies under specific conditions. So such as if we supplement growth factors or any supplementation or any drug treatment, So what is the nature of cell, or what are the number of viable cells which can form the colonies? So in this, cells are plated at low density in a culture medium and allowed to grow for a definite period in the and incubated. So when we know initial number of cells, okay, initial number of cells, and after a certain period of time we will count the final number of cells. So we now know that only how many numbers of viable cells are there which were capable of forming colonies. So इसका ये जो टेक्निक है, इसका जो यूज़ है वो एंटी कैंसर ड्रग के एफिकेसी में अगर हमें मेजर करना है, सो वी कैन यूज़ टू एसेस द एफिकेसी ऑफ एंटी कैंसर ड्रग्स, रेडिएशन सेंसिटिविटी ऑफ ट्यूमर सेल्स, study the effect of genetic mutation on cell proliferation and survival. So these are the some application of this technique. Now last technique is ELISA. The full form is enzyme linked immunosorbent assay, ELISA. So ELISA is widely used immunoassay technique for the detection and quantification of antigen, antibodies, protein, peptides, hormones and other biomolecules in the biological sample. So it relies on the specific binding of the antigen to the or the antibody to on a so it relies on the specific binding of an antigen or antibody to capture molecule immobilized on a solid surface. So it follows detection using enzyme linked secondary antibodies. Colorimetric assay is used here. So what is the procedure? The sample is added on the microplate, okay, coated with either, for example, we have coated it with antigen, specific antigen, which is specific to our targeted analyte. So after incubation we wash to remove unbound molecule. So now enzyme linked detection antibody is added. So what we will add? An antibody containing the enzyme which will react to the targeted analyte. So this enzyme catalyzes a colorimetric reaction upon addition of the target analyte, which is also can be called substrate, So the intensity of the signal, जितना ज़्यादा signal intense होगा, जितना ज़्यादा color आएगा, उतना ज़्यादा it will mean that the signal is proportional to the concentration of the target molecule in the sample. For example, जितनी ज़्यादा intensity उतना sample में आपका particularly effective biomolecule is present in the target sample. So ये किस में यूज़ होता है? फार्मास्यूटिकल डेवलपमेंट में यूज़ होता है, फूड सेफ्टी टेस्टिंग में यूज़ होता है, और डिजीज़ डायग्नोसिस में, बायोमार्कर डिटेक्शन में, ड्रग स्क्रीनिंग में, इन सब में यूज़ होता है. तो आई थिंक अब इसमें से कोई भी क्वेश्चन आएगा तो होपफुली सॉल्व करना आसान होगा. So the next question is, which is the proper order of steps in the northern hybridization? So the answer is R in last D option. So why the answer is D option? Let's discuss first what is northern hybridization. So northern hybridization is a hybridization technique which is used to detect RNA in the samples. So first step, because we have to see whether our targeted RNA is present in the sample or not. So first we have to isolate this RNA from the sample. So first step is always isolation of the RNA And because the sample can contain multiple RNAs, variety of RNAs, so the targeted RNA which we have to target, so we have to first separate the RNA. So for this we have to use electrophoresis. After electrophoresis, what happens is, because RNA gets separated on the basis of their sizes, so we get multiple bands. So but for example, we have to detect this RNA in the sample, and this is our target of interest. So we have to hybridized, because mRNA, for example, this is mRNA, and mRNA is always single stranded. So to find out or to detect the mRNA in the sample, we have to label it with a probe. Okay, so for that, let us discuss from the start. RNA samples are separated by gel electrophoresis. Okay, then after electrophoresis, the RNA molecule in the gel are transferred because these mRNA are still on the gel. So from this gel, the RNA molecules are transferred onto a solid support such as nitrocellulose membrane. Then membrane is then incubated with labeled RNA probe. मतलब कि वो RNA जो इस mRNA का कॉम्प्लीमेंट्री होगा और इससे बाइंड करेगा, बट ऑल्सो दैट mRNA कॉम्प्लीमेंट्री RNA इज प्रोब्ड. तो ऐसा mRNA हम यूज़ करते हैं. दिस इज कॉल्ड प्रोब लेबलिंग. सो we have hybridized this mRNA with the probe mRNA. So after the incubation period, washing is done to unbound the probe, and the membrane is exposed to the washing is done to remove the unbound probe, and then the membrane is exposed to the X-ray films or to radiography to visualize the interested targeted RNA with hybridized probe. So the northern hybridization step will be RNA isolation, electrophoresis to separate the RNA, probe labeling to visualize the RNA, and autoradiography to see the targeted RNA bounded with the probe RNA. So northern hybridization is used to study gene expression by detecting and quantifying the levels of specific RNA transcript in the cells or tissue. अब अगर प्रैक्टिकली देखा जाए तो ये कैसे होता होगा, ये देख लेते हैं. For example, there is a plant and we have given a particular stress, for example salt stress. Okay, and after a while we collected the sample of those plant which have been subjected to the stress, and these mRNA, whole mRNA from the plant leaves have been isolated. The first step is RNA isolation from the leaves. Then, because there may be multiple RNAs and all RNA have different functions, but we are mostly interested in a, for example, particular metabolite forming RNA, for example X metabolite forming RNA. So we have to separate with the help of gel electrophoresis. After the separation, नाइट्रोसेल्यूलोज मेम्ब्रेन पे ट्रांसफ़र किया जाएगा सैंपल. देन प्रोब लेबलिंग करी जाएगी, एंड देन ऑटो रेडियोग्राफी करी जाएगी. तो वी कैन कंपेयर दिस एक्स मेटाबोलाइट RNA with the help of auto radiography, where we can compare a non-treated control plant if the band is thin, and in the treated plant if the band is thick, that means the stress has made the plant or induced the plant to produce this X metabolite RNA more. That is why it is present in the larger amount in the sample. That is why band is thick, or during the visualization we can say there is high rate of production of this mRNA.